Kalau nilainya kaki beda ya tulisan. Oh,
So you're still thinking of me, just like I know you should. I cannot give you everything, you know I wish I could. I'm so high at the moment, I'm so caught up in this. Yeah, we're just young, dumb, and broke, but we still got love to give. While we're young, dumb, and broke. Young, dumb, young, dumb, and broke. Young, dumb, and broke. Young, dumb, broke, high school kid. Just a young gun with a quick fuse. I was uptight, wanna let loose. I was dreaming of bigger things and wanna leave my old life behind. Not a yes sir, not a follower. Fit the box, fit the mold, have a seat in the foyer. Take a number. I was lightning before the thunder. Thunder, 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 feel the thunder, lightning and the thunder, thunder, feel the thunder, lightning and the thunder, 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 thunder. We have so much in common. We argue all the time. You always say I'm wrong. I'm pretty sure I'm right. They say you're basic, they say you're easy. You're always riding in the backseat. Now I'm smiling from the stage while you were clapping in the nosebleed. Young, dumb, and broke. Young, dumb, and broke. Young, dumb, and broke. Young, dumb, broke, high school kid. Thunder, feel the thunder. Lightning and the thunder. Thunder, feel the thunder. Lightning and the thunder. Thunder. Young, dumb, lightning and the thunder. Feel the thunder.
Already study program including law, general, medical education, and chemical engineering study program. The whole study program has been accredited.
Unicel, awesome.
Yeah, then now we also have hundreds of alumni who have worked in various
Unicel, awesome.
companies
Awesome.
and in dozens.
Unicel, awesome. Bye.
And also we have collaboration from
Awesome!
and also we have
Join us. Unicel, awesome.
so many
Wait. We're not done yet. You want please?
within and outside country. Finally, postgraduate program continue to prepare for the online learning system in the
Thunder, 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 feel the thunder, lightning and the thunder, thunder, feel the thunder.
especially to minimize the impact Therefore, this activity is a form of scientific research. Okay.
Hai, saya Anin.
Hai, saya Fauzan.
Hai, saya Zima.
Hai, saya Din.
Hai, saya Ferry.
Kami dari
Biro Pendaftaran dan Penginapan.
Watch this.
Tama bro.
Cak ini sekarang.
Oke, barangkali bisa kita mulai. Studium, silakan. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Waalaikumsalam warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
A very good morning and a warm welcome we deliver to our guest of honor, Dr. Noraza Muhammad Nawawi from Center Graduate Study of University Selangor, Malaysia. The Excellencies, Dr. Sri Rahayu, Magister Management, the Director of Graduate Program of Muhammadiyah Palembang, Eko Arianto, Magister of Chemical Engineering, PhD, the Secretary of Graduate Program of Universitas Muhammadiyah Palembang, Dr. Sri Wardani, Magister of Science, the Head of Biology Education Study Program of Graduate Program of Universitas Muhammadiyah Palembang, and all webinar participants and attendants, ladies and gentlemen, good morning.
Morning.
And welcome to International Studium Generale Microbiology Industry, organized by Biology Education Study Program of Graduate Program of Universitas Muhammadiyah Palembang. The Excellencies, ladies and gentlemen. First of all, let's praise the Almighty God for His blessing. We are able to attend this International Studium Generale Microbiology Industry. Salawat and salam may always be poured out to our Prophet Muhammad Sallallahu Alaihi Wasallam, his family and companions. All right, to start this opening ceremony, let's say Basmalah. Bismillahirrahmanirrahim. Now we go on to the reciting Al-Quran by Bapak Kadir. The time is yours.
Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
A'udzu billahi minasyaitonir rajim. Bismillahirrahmanirrahim. Ya ayyuhan nasu inna khalaqnakum min dzakarin wa unsa Ja'alnakum su'uban wa qabaila lita'arafu. Inna akramakum indallahi anfusikum innallaha alimun khabir.
qalatil a'rabu amanna qul lam tu'minu walakin qulu aslamna walakin qulu aslamna walamma yadkhulil iman fi qulubikum wa in tuti'u Allah wa rasulahu la yalikum min a'malikum syai'an inna Allaha ghafurur rahim inna Allaha ghafurur rahim. Sadaqallahul azim. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you to Bapak Kadir for reciting Al-Quran. May Allah bless all of us. Amin ya rabbal alamin. Now we go on to sing the national anthem Indonesia Raya. Ladies and gentlemen, please stand up. Bangsa dan marilah kita bersatu Indonesia tanah rakyatku semuanya marilah jiwanya badan untuk Indonesia Raya. Indonesia Raya, merdeka, merdeka, anakku negeriku yang kecil. Indonesia merdeka, Indonesia Raya. Indonesia Raya, merdeka, merdeka, negeriku yang kecil. Indonesia merdeka, merdeka, Indonesia Raya. Thank you, ladies and gentlemen. Please have a seat. The Excellencies, ladies and gentlemen, to begin the opening ceremony today, here is the report from the head of graduate program of Universitas Muhammadiyah Palembang. Ibu Profesor Sriwadani, Magister of Science. Times is yours.
Thank you, Ms. Dian. Assalamualaikum warahmatullahi wabarakatuh.
Assalamualaikum warahmatullahi wabarakatuh.
Your Excellency, Bapak Eko Arianto, Magister Chemical Engineering, PhD, the Secretary of Graduate Program, Universitas Muhammadiyah Palembang, our distinguished guests, and also our keynote speakers from Center Graduate Study, University Selangor, Ibu Norazah Muhammad Nawawi, PhD, and our beloved or participant who participate on International Studium Generale webinar. First of all, I would like to say all praises to Allah Subhanahu wa Ta'ala who have given His grace and favor to all of us. Thus, we can meet virtually at the International Studium Generale this morning. And salawat and salam may always be poured out to our Prophet Muhammad Sallallahu Alaihi Wasallam, his family, and the companion. Thank you to ladies and gentlemen for your participation. Currently, microbiology research is widely applied in the industrial world. This development is certainly an interesting topic to discuss. But for the general public and the industrial community, for lecturers and teachers, and particularly for graduate students in Biology Education Study Program of Universitas Muhammadiyah Palembang as enriched material on Advanced Microbiology course. The topic of the webinar today is microbiology application from academic exploration to industrial implementation. It's a reflection of the issue research, and it could hopefully provide a new horizon for all of us. Okay, ladies and gentlemen, as the chief executive, I would like to report several things with the activities today. The number of participants who have registered this webinar are 197 people, which are consisting of lecturer, middle and high school teachers, students and alumni from various education institutions in Indonesia, such as from South Sumatra, Jambi, Lampung, Jakarta, Yogyakarta, West Java, Central Java, Sulawesi, and some people from Malaysia and present United Kingdom. We hope this webinar can provide a broader insight into microbiology and in implementation in our life, particularly for industrial world as the application of various research which have been carried out. We also hope this activity continuously will be carried out regularly with various different topics regarding the development of biological science and the development of other biology education. Okay, dear ladies and gentlemen, let me introduce more about Biology Education Study Program of Graduate Program Universitas Muhammadiyah Palembang. Izinkan saya untuk memperkenalkan lebih jauh mengenai program studi pendidikan biologi S2 Universitas Muhammadiyah Palembang kepada para peserta. Program studi pendidikan biologi S2 UMP berdiri sejak tahun 2015 yang merupakan satu-satunya program studi pendidikan biologi di Sumatera Selatan. Pada tahun 2017 telah mendapat pengakuan berupa akreditasi dari Badan Akreditasi Nasional Perguruan Tinggi (BANPT) dengan nilai B, di mana jumlah total mahasiswa sampai saat ini adalah 86 orang dan telah menghasilkan lulusan sebanyak 53 orang. Alumni dan mahasiswa yang ada saat ini itu rata-rata sudah bekerja dan sebagian besar berprofesi sebagai guru dan pegawai. Sisanya ada juga yang belum bekerja dan sebagian adalah fresh graduate. Dosen tetap semuanya bergelar doktor dengan bidang keahlian biologi dan pendidikan biologi, serta berbagai bidang ilmu lain yang relevan dengan biologi, di mana pendidikan biologi ini dalam perkuliahan disampaikan di dalam 3 bahasa, yaitu bahasa Indonesia, bahasa Inggris, dan bahasa Arab. Adapun prestasi yang pernah dicapai oleh program studi pendidikan biologi antara lain mendapatkan prestasi sebagai program studi S2 terbaik berdasarkan hasil dari audit mutu internal Universitas Muhammadiyah Palembang pada tahun 2017 dan tahun ini 2022, di mana pada tahun 2022 ini juga program studi pendidikan biologi menempati 10 besar program studi terbaik berdasarkan Sinta Skor dalam 3 tahun terakhir. Untuk beasiswa, alhamdulillah program studi pendidikan biologi menyediakan berbagai beasiswa, diantaranya ada beasiswa bagi fresh graduate S1 dari program studi pendidikan biologi Universitas Muhammadiyah Palembang dan juga dari Universitas Islam Negeri Raden Fatah Palembang. Karena antara 2 lembaga ini terdapat kerjasama. Kemudian juga beasiswa bagi mahasiswa yang berasal dari negara Palestina. Kemudian beasiswa LPDP untuk penelitian tesis dari Kementerian Keuangan. Kemudian ada beasiswa program studi untuk mahasiswa yang berprestasi. Nah, pada kesempatan yang baik pada hari ini, kami juga berharap bagi para peserta, khususnya bagi guru IPA SMP dan guru biologi SMA di Sumatera Selatan, dapat melanjutkan studi strata 2-nya di Pendidikan Biologi Universitas Muhammadiyah Palembang. Okay, the last but not least, I would like to say many thanks to keynote speakers, Ibu Norazah PhD, who has pleased to share her knowledge with all of us here. I really hope that the cooperation and friendship between Universitas Muhammadiyah Palembang and University Selangor still continue in the future. Hopefully, this webinar bring many benefit to all of us. I also hope we are always in good health and always the protection from Allah Subhanahu wa Ta'ala. Okay, thank you and sorry for any shortcoming. Nasrul minallah wa fatun qorib. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you, Ibu Sri Wardani, for the report. Now we move to welcome speech by the Director of Graduate Program of Universitas Muhammadiyah Palembang. And this chance will be delivered by Association Bapak Eko Arianto, Magister of Engineering, Chemical Engineering, PhD. Time is yours.
Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh.
The Honorable Ibu Dr. Sri Wardani MSc, as Head of Biology Education Postgraduate Program, and also Ibu Norazah Muhammad Nawawi, PhD from University Selangor, Malaysia, as a speaker today. I apologize from Ibu Sri Rahayu because she was unable to give the speech at today's at the meeting today, and she give me opportunity to give a speech at the opening ceremony today. Praise and gratitude to Allah Subhanahu wa Ta'ala, who has given countless blessings so that it strengthens our determination to take part in the Studium Generale of Biology Education Study Program. Salawat and salam may continue to be brought out to the Lord of the Prophet Muhammad Sallallahu Alaihi Wasallam, who has always been guide us in this world and the hereafter. May we all get intercession until last day. Amin ya Rabbal Alamin. Distinguished guests, all the participants. Currently, the postgraduate program at Universitas Muhammadiyah Palembang has already 4 study program, including law, management, biology education, and chemical engineering study program. So all of this study program have been accredited from BAN PT. Then at this time, we also have almost 1,000 of alumni who has worked in various companies. Yeah. And also, it doesn't stop there. We have now established cooperation from national or international. And currently, the postgraduate program continue to prepare for the learning system in the new era. We realize that the impact of the COVID-19 pandemic is very significant, especially to minimize the impact of the learning loss. Therefore, this activity is a form of scientific development in microbiology subject. Yeah. Hopefully, this activity will provide a good benefit for the participant. So this our hope, yeah. Especially in our research and learning process. So hopefully this activity can be starting point for realizing the internationalization of study program at Universitas Muhammadiyah Palembang. So finally, I open this activity I open this ceremony, I open this Studium Generale by say Basmalah. Bismillahirrahmanirrahim. That's all I can say. I apologize for any mistake and shortcoming. So for the closing opening speech today, I try to give us a pantun, but using bahasa. Jalan-jalan ke kota Padang, singgah sebentar membeli tali. Jika ada kesempatan, kami tunggu di pendidikan biologi. Billahi fisabilillah, wastabiqul khairat, wassalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Bapak Eko Arianto, for welcoming speech. So, by the end of welcoming speech by Bapak Eko, it means that we have now come to the end of this opening ceremony. So, ladies and gentlemen, let's close this opening ceremony by saying Hamdalah. Alhamdulillahirabbil alamin. The Excellencies, ladies and gentlemen, so it's time for International Studium General Microbiology Industry. It is delivered by Ibu Noraza Muhammad Nawawi. But before I will I'm going to tell you the curriculum vitae of our keynote speaker today. Well, we go on to and I'm going to recite the curriculum vitae from Ibu Noraza Muhammad Nawawi. She was appointed as a lecturer at University Selangor since 2004, and she has taught various subjects and supervised many students. In research, she has been coordinating and maintaining microbial culture collections in Institute Bio IT, Selangor, since the date of its establishment. Among various microbial cultures in Institute Bio IT collection, Rhodococcus has been recognized as a potential strain for numerous applications, including bioremediation, pharmaceutical, and chemical industries. As a microbiologist, she is investigating the capacity of both full and immobilized cells on panel biodegradations. Besides Rhodococcus, she currently involved in a project to investigate the potential of locally isolated microalgae and other types of bacteria for natural phagocytosis. Currently, she also awarded a national grant focusing on synthesizing silver nanoparticles by microorganisms for agricultural applications. Now, let's do academic qualifications. Doctor of Philosophy, Biotechnology in UPM Malaysia, 2016. Master of Science, Genetics in UKM Malaysia, 2004. And Bachelor of Science, Molecular Biology, Microbiology in UKM Malaysia, 1999. She has current professional memberships, National STEM Association, NSA. The position, she is secretary from 2019 until to date. She is Malaysian Society for Molecular Biology and Biotechnology, MSMBB. She is a member 2021 until to date. And then she is the member of Malaysia Society for Microbiology, MSM, in 2018 until to date. And her previous employment, she is the lecturer of University Selangor, begins in 2004 until 2010. She is Malaysia Agricultural Research Institute. She is a research officer 2003 until 2004. And she was research assistant in University Kebangsaan Malaysia 2000 until 2002. She has several conferences and training. Training on molecular biology techniques, training on genomic and molecular biology evaluation, Secretariat International Conference on Life Science Revolution 2016, Director Women and Science International Seminar, Treasurer International Conference on Life Sciences Revolution 2015, Mentor Minty STEM Program, First National STEM Program Seminar, Third International Conference on Biosciences, 2019 Classroom Teaching Research for All Students Conference, CTRAS, Third International Conference on Molecular Biology and Biotechnology 2019. Next, Persidangan Kebangsaan Pengajian Asasi dan Umum Persepadu 2019 and National Per-University Seminar 2019. She has 9 research projects from 2006 until 2023, and then she has 2 publications in 2016 and 2021, and then she has several publications from 2014, if I'm not mistaken, until now, 2022. So that's the curriculum of our keynote speaker. And now this Studium General will take 90 minutes for delivering speech by Dr. Noraza Muhammad Nawawi. And after that, we have 30 minutes for question and answer sessions. And then we have closing this webinar. All right, so we go on to Studium General Microbiology Industry by Dr. Noraza Muhammad Nawawi. Kepada Ibu Dr. Noraza Muhammad Nawawi.
Ya, Assalamualaikum warahmatullahi wabarakatuh.
Warahmatullahi wabarakatuh.
Good morning to all. The Honorable Dr. Iko Arianto, the Secretary Graduate Program University Muhammadiyah Palembang, and Dr. Sri Wardani, Head of Biology Education Study Graduate Program University Muhammadiyah Palembang. So first of all, I would like to thank the organizer for inviting me as the speaker of today's session. And we will discuss or sharing some information related to microbiology. I was told that the participants comprise of a different level of students as well as teachers in biology field. So I hope what I will share today, we will get, the participant will get benefit from it, and we can plan further action related to microbiology. So before we start, let me share the slides. Okay, the topic that was suggested by the organizer is related to microbiology application. So today I would like to share some general study, as well as what we have done in University Selangor from academic explorations, starting from getting or collection data for knowledge, and also planning toward the industrial implementation. Thank you to the Master of Ceremony. Just now read my CV. So that's basically my humble background. and currently I am the Deputy Dean of Academic at Center for Graduate Study, University Selangor. So I received this invitation based on my humble experience in microbiology, okay, starting from my bachelor degree, Bachelor of Science, purely in microbiology. And at this stage, I did some research related to photosynthetic bacteria, isolating photosynthetic bacteria for petroleum degradation or hydrocarbon degradation. So since then, my interest in microbiology increased, and I further my study for Master in Science. And during my master, I'm focused on fungus, and at that time, I interested to do some molecular work to study the promoter of genes that code for cutinase, which is the responsible enzymes that cause pathogenicity for the fungus, which known as Glomerilla cingulata. Moving on to my, after that I worked as a research assistant and also lecturer. Then I did my PhD, and this time, because of our collaboration, I will mention later, I did some research related to valuable bacteria and focus on bioremediations. So inshallah, with that background, with that experience, I will share about the research did by me, myself, and also my team at UNICEF. So basically, before we discuss about research in detail, we will do some introduction about microbiology, just a recap related to microbiology. Maybe there are some participants that are not familiar with this field. We will see or recap the applications or the importance of microbiology field, and moving on to the related research. And also I will introduce some or the postgraduate program offered by University Selangor. Okay, before we go further, can anyone tell me what is this figure about? Any participant can give your feedback.
They are bacteria.
What is this picture about? Can you guess?
Basil bacteria.
Ibu Marina said it's a bacteria. Yes, but can you guess at what surface?
Basil.
Sorry?
The basil bacteria.
Basil bacteria. Yes, it's a bacillus bacteria. And actually, this figure shows you bacteria, a rod-shaped bacteria on the head of a household pin. So you just imagine, only at the tips of the pin, we can observe thousands of bacteria. Okay, this figure under scanning electron microscope, so we can see the rod-shaped bacteria in yellow color. So Yes, so microbiology, microorganisms are everywhere, even on the head of a pin, also we can see thousands of bacteria. Okay, when we talk about microbiology, okay, maybe some people might feel it's interesting, or for certain people maybe feel that this field is hard, okay, membosankan, because we cannot see the microorganisms using our naked eyes compared to plant or animals. So doing research in microbiology need your passion and need your concentration, okay, in order to get a good and correct results. So I hope by the end of my sharing, okay, it can trigger your interest in this field, okay, or to renew your interest or your feelings toward microbiology field. Okay, believe me, yes, I agree that we need concentration, we need patience in doing research related to microbiology. However, when you get the results, okay, you can produce the results, okay, you will feel the satisfaction. And at that stage, you feel that you want to share to everyone, okay, everyone in the world that you get, okay, after all the constraints and difficulties. Okay, let's recap some point about microbiology. So basically, microbiology is the study of microorganisms and their activities. So what is microorganisms? Okay, in Bahasa Melayu, normally people always call it as kuman. Okay, kuman. Okay, actually it's not a good definition, because kuman normally refer to a bad microorganisms. Okay. Whereas microorganisms, yes, they are small, we cannot see with our naked eyes, but there are some bacteria that give benefit to us. So saya masih ingat semasa saya mula-mula belajar degree dulu, okay, ramai yang kata, kenapa you nak study pasal kuman ni? Okay, tak ada bidang lain ke nak belajar? Okay, so but I teruskan juga, okay, supaya saya nak buktikan bahawa microorganisms itu, microbiology itu bukan kuman, bukan menjijikkan, but it is interesting. Okay. So that's about, so we need a microscope or magnifying apparatus to observe the microorganisms. So microbiology are wide field, okay, because microorganisms are present everywhere, even on us as a human or in our environment. Okay, they are relevant to us. Okay, they influence our life. Okay, either beneficial or as well as detrimental. And they are indispensable component of our ecosystems. Without them, our ecosystem will perish, and the microorganisms also have harmed humans and disrupted society over the millennia. Yes, we agree on that, especially throughout the pandemic seasons that we gone through last 2 years. Okay, we agreed on this, but there are vast scope in the field of microbiology due to the advancements in the field of science and technology. So there are a lot of knowledge in microbiology that we have to find because I just share some interesting facts about microbiology. Okay, our body has more microbes than there are human cells. Means there are more microorganisms compared to our cells. And the human mouth has approximately 500 bacteria species, not cells, but species. So one species will have much number. And half of the oxygen in the atmosphere is generated by microbes. So they help us, very important for us in our daily life. So either, though microbes are small, okay, we cannot see using our naked eye, but they have complex processes, structure, and behavior like other organisms. Okay, when you study further about bacteria, fungi, virus, or algae, for examples, okay, then they have their own behavior to sustain and give their functions. And it is true that there are microbes everywhere, but not all microbes are everywhere. Okay, means not the pathogenic and non-pathogenic. They have their own interests. They have their own hosts. So they are not all microbes are everywhere. And believe it or not, microbes can be used to solve a crime. Can anyone comment on this? Are you agree? How microbes can be used to solve a crime?
Yes.
Can you relate with any point in microbiology? Okay, we will see later how microbes can solve criminal cases. So from what we discussed just now, we can see the scope in this field is immense due to the involvement of microbiology in many fields. Okay, for example, in medicine, pharmacy, dairy industry, clinical research, water industry, agriculture, chemical technology, as well as nanotechnology. Okay. A new field which involve microbiology. So why microorganisms or microbiology important? Okay, because we can investigate the potential of microorganisms. Okay, maybe for example, fighting microbes to get a good medicine for certain diseases. Okay, for example, during the COVID-19 pandemic, okay, everyone know that the process of vaccine development in order to cure or to avoid or to protect us towards COVID-19 viruses. So the more we know about microorganisms, the more the quality of human life is improved. So that's why research in microbiology never end. It still continue, especially with the development of science and technology. So there are various types of microorganisms. We have bacteria, we have fungi, we have viruses. Though there are a conflict whether we can put virus as the microorganisms because they need hosts, they not live organisms, but because they cannot see using our naked eyes, so they was classified under microorganisms. So there are advantages and disadvantages of microorganisms. Basically, in our daily life, okay, we consume microorganisms as our food and beverages. Okay, we Asian, especially Malaysia and Indonesia, especially Javanese, okay, always eat tempeh. One of products produced by fungi. Okay. Kadang-kadang bila kelas microbiology, kalau ada pelajar yang tidak tahu, mereka macam tak sangka, okay, they can't believe it, they eat fungi. Okay, daripada kecil dia makan, dia tak tahu that's the kind of microorganisms. Okay, so mereka akan lebih tertarik untuk belajar morfologi dan sebagainya. Okay. Our kids normally will eat cultured drinks. Okay, for example, Yakult or whatever. Okay, actually in the cultured drinks there are billions of bacteria, and it gives benefit for our digestive system. Bacteria also can give or act as degrading agent, especially in soft pollutant problem, okay, where we call it as bioremediation. Okay, we also cannot deny that microorganisms give disadvantage. They cause diseases such as AIDS, chickenpox, okay, mumps. Okay, these all common disease. But nowadays, people more alert about microorganisms, okay, since the COVID-19 pandemic. Microbes also cause teeth decay and also food poisoning if we do not take precautions during our food consumptions. Just share one picture, a drawing of microbes. This was done by Arthur Hill Hessel. He saw the morphology under microscope. Okay? He took samples of water from River Thames at different locations, and he able to identify many microscopic organisms, okay, not perceptible to the unaided eye. Okay, so different locations give different morphology. Just share one tree of life that shows different types of microorganisms. Actually, the process was started in 1866 by Haeckel, who proposed the creation of third kingdom, the Protista, which include algae, protozoa, fungi, bacteria, and viruses. In 1957, Stenier divided the protists into eukaryote and prokaryote cells. Okay, we always have to remember the difference between prokaryotic and eukaryotic cells. When you are a microbiologist or learn about biology, you cannot run away about prokaryotic and eukaryotics. Yeah, I still remember during my, just share some experience. Okay, during my oral presentation, the viva for my masters. Okay, I prepared all the data about my experiment, and suddenly the examiner asked me, "Can you tell me the difference between prokaryote and eukaryote?" Okay, masa tu macam terlupa, because kita fikir yang lebih advance, padahal prokaryote and eukaryote tu sangat penting in order to relate the characteristic of microorganisms. So microbiology currently divided into bacteriology, virology, parasitology, and mycology. Okay, depending on the type of microorganisms. Okay. In studying microorganisms, we cannot run away about classification. Not only microbiology, okay, when we study about biological organisms, we cannot run away about the classifications. We have to remember from the domain up to the species. So this is the classification of E. coli. Okay, just a figure of a microbiologist. This is a microbiology officer aboard a US naval ship who examines the wound cultures in the ship's microbiology lab. Okay. So microbiologists can work everywhere. Okay. Not limited in university, but they need microbiologists in industries, also in defense. Okay, when we talk about applied microbiology, okay, we cannot run away with fermentation. This is one of the oldest and well-known examples of applied microbiology. In the picture, you can see the large tanks. Actually, it used to ferment the fermentation of grapes to make wine. Okay, actually, the applied microbiology or the industrial microbiology has started since decades ago. Okay, because the Asian also drinks wine, and without knowing that wine is processes and you using microorganisms in the process, okay, either using bacteria or yeast. So in industrial microbiology or application microbiology, this man, Louis Pasteur, is one of the founders of microbiology. Okay. His finding help us until today, okay, the techniques to minimize or to sterilize our apparatus during research. So this is a set of experiment done by Pasteur, and he tested of spontaneous generations. Another man, Robert Koch, a pioneering microbiologist. He responsible in doing research and methods that help to link the causal nature of microbes to certain diseases, including anthrax. So he give ideas and help us until now on microbiology. Okay, Anthony Leeuwenhoek, okay, we cannot say his finding not important because he is the first scientists that use microscope and identify the microorganisms. So until now, starting from Leeuwenhoek, and until now, many scientists involved to study techniques to know the characterization or to identify the morphology. Okay, gram stain is one of the techniques where we can differentiate between gram positive and gram negative bacteria. Okay, so if you are a microbiologist, you have to remember about gram stain. So which one is positive, which one is negative. So it based on colors. To me, how I remember is if I got weak results in or fail in my study, okay, normally teacher will write in red, right? So to me that is negative. So I remember negative is red and positive is blue. Okay, move on to the relationship with humans. Okay, we as the human are sterilized in the utero of our mother, but shortly after birth, microorganisms will colonize. Okay, so it will start to grow and dominate our body, okay, to all of parts of human body. So various type of microorganisms can be isolated from our body. So we call it as microbiome. So I move on to the application of microbiology. We already discussed about the character and also the application of microbiology just now. Let's see discuss in details about it. So in addition to studying the natural history of microbes, so it deals with every aspect of human microbe and environmental interaction, because during the interaction, it includes ecology, genetics, metabolisms, infection, disease, chemotherapy, immunology, engineering, industry, and also agriculture. Okay, we also do not know exactly when the bread, the production of bread or cheese or yogurt started. Okay, so because it already used by the ancient time, okay, but using the technology, until now we know that in production of bread or cheese or yogurt, okay, they need microorganisms as the one of the ingredients. Okay, for bread, for example, the bacteria will act with the flour and sugar and produce carbon dioxide and oxygen. So from microorganisms, there are many important products can be isolated or can be produced by the microorganisms. Okay, let's see scope the importance of microorganisms. First, let's see in our environment. Okay. Besides involved in degrading organic and inorganic materials, they also play important roles in recycling, especially in carbon, nitrogen, and sulfur cycles. Okay, so without them, without microorganisms, our biosphere is not stable. Okay, because these cycles is important to maintain the ecological balance on our Earth. In the environment also, the microorganisms not only act individually, okay, they act in symbiotic relationship, okay, especially to maintain the soil fertility or to do bioremediation process. Okay, some microorganisms also cause diseases. Okay, they are plant or animal pathogen, but others can act as biological control agents against the disease. So they will help to maintain the food chain and food web to balance our ecology. Besides that, some microorganisms also spoil our food and deteriorate materials like the iron pipes, glass lens, computer chips, jet fuel, paints, concrete, metals, plastic, paper, and also wood filings. Okay, so we can say that every part of our life, there are microorganisms involved. So today we know Gretzky is the scientist who discovered about nitrogen fixing bacteria. Okay, microorganisms widely spread in different environment, either in extreme conditions. So these figures shows you microorganisms in a cold environment, the ice algae in Antarctica. Okay, I have one friend who currently do research in Antarctica. She just came back from Antarctic, and she able to isolate various kind of microorganisms, bacteria, fungi, and also algae, and she can see potential of those microorganisms in industries. Okay, beside cold, microorganisms also can be isolated from hot environment. This is, you can see the algae growing in a hot pool in New Zealand. This slide shows you a fungi as decomposer. Okay, because they are important to in the nutrient cycles. Okay. Basically, these fungi grow on the side of tree, okay, and they receive their nutrients through their hyphae and invade and decay the tree trunk. So it helps to decompose the tree trunk. A figure about cyanobacteria, or also known as blue-green algae. Okay. One of the candidates that I also trying to isolate from our local environment. And also many people, many researchers do research on cyanobacteria, especially in related to biofuel. Okay. You can see here, this is a blue-green alga culture in a specific media. Okay. Green in color. This cyanobacteria can help in agriculture to fix atmospheric nitrogen to soils. That's why a lot of research study related to biofuel. This figure or the image shows you the biotrophic relationship between a glomerulus mycota and the roots of a plant. Okay. So you can see the symbiosis between microorganisms and plant. And this image of photosynthetic fossil cyanobacter in a billion-year-old rock formation of Glacier National Park, Montana, USA. So microbes are everywhere. Different environment will produce different or will enhance the growth of different type of microorganisms. Okay, so beside in environment, so we can say that yes, we could agree that microorganisms important in the environment. We can see the application of microbiology in environment. Okay. Now we move to the application of microbiology and medicine. Microbiology cause diseases, for example, smallpox, okay, cause variola virus, cholera by bacteria, Vibrio cholera, and also malaria caused by protozoa, Plasmodium, and many more diseases. So from
biasa sekarang fotografi tidak butuh keahlian
from
karena
the character, okay. So researcher did various kind of techniques to find important drugs in order to kill or avoid the growth of microorganisms. This is a common techniques of antibiotic testing, where we normally soak this and put it on the lawn of bacteria. And if there is halozone, means the antibiotic or the products or the metabolite can kill the bacteria. When we talk about antibiotic, we cannot run away or we have to recognize the findings by Alexander Fleming. Okay. In medicine also, we can see the development of vaccine for COVID-19. Okay, we have different kind of vaccine, okay, produced by different body, okay, using different technology. Okay. So since 2020, laymen also know about DNA, RNA, okay, mRNA, about polymerase chain reaction, about immune system and everything. So that's our responsibility as a microbiologist to explain more about the doubt of public about the vaccine. Okay, because there are many people still try to avoid to accept vaccine as the precautions. Okay, yes, we do agree a lot of stages, but we have to admit that microorganisms also have their way to protect themselves. So they mutate and getting stronger. So we as the scientists has to think how to create a better vaccine to avoid the virus. Okay, that's for medicine. Okay, in food industry, as we already mentioned earlier, they're important in various type of food, especially in related to dairy and also bread, sauces, and also beside products, it also cause food spoilage. With the development of microbiology and also biotechnology, okay, means with the enhancement of science and technology techniques, okay, we can choose the correct genes and clone into a correct plasmid and host to produce products, to synthesize drugs or enzymes, okay, based on the interest and also demand. So biotechnology help the microbiology field to commercialize or give more products in industry. Okay, we can see the application of genetic engineering to produce insulin. For example, insulin is the is important to control sugar level. Okay, so those people who are not capable, where their system is not capable to produce insulin, so they need to inject insulin. So with the advancement of biotechnology, we're able to clone the gene for insulin and also transform into E. coli, and the bacteria help us to produce a mass amount in insulin, and at the same time can minimize the cost of production. As well as we can get during a short period of time, because bacteria can grow within 24 hours. Okay. Besides insulin, we also have human growth hormone, interferon, and so on. Okay. Okay, another applications of or the importance of microorganisms in biotechnology is this bacteria or Thermus aquaticus, okay, which release tag polymerase enzymes, okay, that very important in polymerase chain reaction to multiply the number of genes or part of important genes, okay, which help us to clone the specific genes and produce the products. For example, the important industrial enzymes. At the same time, it also helps us for DNA profiling. So this is one invention or findings that help the to solve their criminal cases. Okay, not only criminal cases, but also in identification. We can see the application of PCR during our pandemic. Okay, a lot of RT-PCR to amplify or to prove that the person already infected by the virus. Okay. Me myself also have experience. I feel that I have a sore throat, and then I'm not able to use the kit to detect. So I proceed with PCR, and yes, from the results, wrote that the COVID-19 is detected from my samples. So I can see the application of PCR in detection of COVID-19 cases. So microbiology can involve in many research, although the microorganisms has a simple structure, so they're easy to study, and it can be the cell factory to produce industrial products, okay, because it can grow quickly and also get the products in a short period of time. So overall, we can conclude the applications of microbiology. Okay, we can see related to genetics because it mainly involves the engineered microbes, okay, to make hormone, vaccine, antibiotics, and many other useful products, okay, for us as the human being. In agriculture, we can see the influence of microbes, okay, either in helping to fertilize the soil, but also in prevention the disease. Next, related to food science, where it involves to prevent the spoilage of food and also foodborne diseases. And we can see the use of microorganisms to produce food and beverages. In immunology, where the study about immune system, okay, where our body need or protect from the pathogens, okay. The nearest example is COVID-19. Okay, everyone know about it. Medicine, where we deals with the identification of plants and measure the cure diseases of human and animals which are infectious to human and animals. In industry, it involves microbes to produce antibiotics, steroid, alcohol, vitamins, and amino acid, insulin, enzymes, and everything with the help of microbes. In agriculture, beside combat the plant disease, we also can use to substitute the chemical pesticide, okay, using a bacterial or viral pathogens. In ecology, we already discussed about it involved in biochemical cycles. In food and dairy microbiology, beside products, it also involves the prevention of microbial spoilage, such as botulism and salmonellosis. So that's why people who want to sell food, okay, they need to get vaccine to avoid any infection. Okay. Industrial microbiology, as we discussed, there is a lot of antibiotic, vaccine, enzymes, and other secondary, primary or secondary metabolites from the microorganisms, okay, can be produced in industry. Microbial physiology and biochemistry is quite similar with the ecology. In molecular genetics or molecular biology, we study a lot of their genes and also the functions and the involvement of microorganisms as the working organisms. And last but not least is genetic engineering. It's arisen from work of microbial genetics and molecular biology actually. So from the informations of genetics, okay, and also the process from DNA to RNA to protein, scientists able to engineer the microorganisms in order to produce valuable products. So as the conclusion about the general information of microbiology, we can see the future of microbiology. Okay, especially to combat diseases, okay, to reduce pollutions and feed the world's populations. Means to produce good food, good beverages, and also vitamins and what so on to increase the quality of life. Okay, if you ever heard about golden rice, for example, that was created using microorganisms in order to solve the ice problem, especially in third world countries. Okay, so a lot of infectious disease can be studied. Okay, how to avoid or to create new drugs or vaccine, especially with the applications of molecular biology and also genetic engineering. Okay, we also able to see the relationship between host and pathogen. Okay, to study how to avoid or how to improve, and we also can study the role of microorganisms as the source of high quality food, and also various kind of enzymes that is important. The role of microbe as pollutant or toxic waste degrader, or we can use microorganisms as the vector to treat disease and also enhance agricultural products. Okay, do I go too fast? Okay, now from the general informations about microbiology, okay, I hope all of us know the benefit of microorganisms in our daily life. Okay, that we as the microbiologists or as the person who know about it have to share with many people about the importance of microorganisms. Okay. Okay. My passion in microbiology, okay, inspired, okay, by a mentor, Emeritus Professor Datuk Dr. Abdul Latif Ibrahim, who always guide and advise us at this university about the importance of microbiology. Okay, he previously he worked at University Putra Malaysia. Okay, and based on his experience in production of vaccine to solve Newcastle disease. Okay. His finding on increasing the productivity and the profitability poultry industries through prevention and control of poultry diseases. Okay, he developed a novel vaccine to control Newcastle disease, and Normally vaccine will distribute through oral or injection, but his vaccine, okay, just through feed, okay, dihasilkan begini, okay, dan dia hanya diberi makan kepada ayam. Jadi all the chickens will be vaccinated. So he's the one among the person who inspired us in University Selangor to do many research related to microorganisms in this university. So basically in UNICEL, we have various kind of microbiological research. Currently, many research focus on biodegradation, such as bioplastic or the toxic pollutants such as dye, phenol, cyanide, and also nutraceutical, for example, cholesterol. So we hope that with these findings, okay, as the academic explorations, it will benefit community, okay, to produce or apply in industry. Okay, it helps to reduce waste, to clean the polluted water, and produce a healthier food products. And at the same time, we also there is group, okay, do about biotransformation related to biofuel and bioplastic. Okay, and the products related to higher value and also sustainable chemicals. In UNICEL, we have one institute of Bio-IT Selangor, led by Dr. Nur Suhaila. So we have various kind of project. For example, it involves Rhodococcus bacteria, and also all the research that I mentioned just now. Okay, so this is related to microorganisms. Besides that, we also have microalgae research. They promote the microalgae growth using natural enhancement medium isolated from the gene area around Malaysia. This group find soil or any ingredients of nature, okay, to grow, to enhance the growth of microalgae. Related to degradations, there is group of researchers do degradation of heavy metals and also degradation of dye. And me, myself, do research related to nanotechnology. Okay, I will show. Okay, one of bacteria that widely used in our research at UNICEL is Rhodococcus. Why we choose it? Because it has broad metabolic diversity and also biotechnological application. It helped to reduce the release of harmful substances into environment. And in Malaysia, when we start this research, there is very little information available in characterization and cultivation of Rhodococcus. So it's part to start the UNICEL Rhodococcus culture program. So Prof. Latif is the one that start the process. So we isolate the Rhodococcus and characterize. And now we do or use the bacteria in many applications. So how we start, I went to various locations throughout Peninsular Malaysia to isolate samples. And from the process, we're able to isolate about 23 Rhodococcus. Okay, and we still keep it and continue the research. So this is the sampling. We target to isolate from polluted area. Okay, and interesting findings that we find that this project is the first in-depth analysis of rhodococcal clade in Malaysia at tropical environment compared to temperate climate. Actually, this project, we have collaborator from University of Cambridge. We use their protocols, but we are unable to isolate using the protocols. It's not effective. We have to modify. And after we modify the protocol, we able to isolate the Rhodococcus. So we believe that because our tropical Malaysian samples was for the previous protocol for post-glacial northern hemisphere samples. But for Malaysia, we have a soil high biological activity or high factor as never glacial. So we have to modify to get the Rhodococcus. So we have 23 until now. We use it. We characterize, and we have already 2 that we do the genome sequences, and we focus on enzymes that have potential in green chemistry for industry and in bioremediations. So just sharing some work related to phenol degrading enzymes. Okay, we try to grow the Rhodococcus on agar with and without phenol, and we can see Rhodococcus able to grow at high concentration of phenol, and then we try to choose which is the best, and this is the molecular approach to screen the phenol hydroxylase gene from the isolates. Okay, and we're able to amplify 1,500 base pair of phenol hydroxylase genes from the strains, and we tested different phenol concentration, and we from the experiment we can see one of the strain able to grow up to 1,500 milligram per liter. So that means it able to use phenol as their carbon source. Okay, then we move or continue the research to immobilize the cells. Okay, so we have a series of experiments to compare the free and immobilized cell, and we can see gel lenggam as the most or the best matrix to immobilize the bacteria, the Rhodococcus bacteria. So we characterize the conditions. There's some figure of the Rhodococcus on gel lenggam bits under electron microscope. We also study about phenol metabolisms, because phenol can be degraded first using phenol hydroxylase, and then it can use either meta or using catechol 1,2 dioxygenase or catechol 2,3 dioxygenase, either by meta or ortho. metabolic pathway. So from the results, I did some clone about the phenohydroxylase and express it. So we able to purify the enzymes to prove that this is the enzymes that responsible in degradations. Okay. This is the results comparing the clone E. coli with phenohydroxylase from Rhodococcus. So we feel that our study related to Rhodococcus is important because it is special for temperate climate. So it's original locally, and then it can continue in future in green chemistry. And also from the research, we develop various techniques to apply either free, immobilized, tracing, and the enzymes from our local selected Rhodococcus in bioremediation. So this is a new knowledge that can be available for further research. So besides phenol degradations, there is one researcher, Dr. Hasdianti Abdullah. She did research on biodegradation of plastic waste using the Rhodococcus. So some of the strain able to degrade plastic. And also he did research on biodegradation of cholesterol. He able to optimize the conditions, what are the genes involved, and also prove that our locally isolated Rhodococcus can degrade cholesterol. Another researcher, Associate Professor Dr. Megalana Lapanmaniam, she focused on dye degradations. Currently, she awarded a grant on bioremediation of batik wastewater because in Malaysia, especially in Terengganu and Kelantan, a lot of batik industry. So she isolate the indigenous microbes from the batik wastewater and then identify, characterize, and also once the process is completed, it can be used to degrade the dye at the polluted river or polluted area. And then for me, I focus on productions or production of silver nanoparticles that can use to kill marine pathogen, and we hope we can use the technology or the products in agriculture industry. Beside other bacteria, our Rhodococcus also proved that can synthesize the silver nanoparticles, and in the process to study in details to kill or inhibit the bacteria that can cause disease on fish. Okay. For example, we use silver nanoparticles to kill Aeromonas hydrophila, and we test on fish, infected fish, and it proved that yes, the Aeromonas cause disease to the fish, and we are in the process to produce or characterize the silver nanoparticles in order to solve these problems. Besides that, I also did do research related to pigment from microalgae, focusing on microalgae isolated from marine and also hot spring because as published by a recent journal, microbial pigments has potential to be used in industries, okay, as the natural food, colorant, textile, antimicrobial activities, and cytotoxic activities. Currently, there are some algae I can see it can produce pigment and in the process to characterize the pigment. This research collaborate with one industry in Malaysia. The previous research, this research also collaborate with industry related to agriculture industry. So basically, this is the aim. We hope we can isolate, we can get a local isolate and produce pigment. In the process to optimize and produce the pigment. So we hope we can achieve what we expect and we can get a product pigment from the locally isolated microalgae. Okay, beside these two, I also have one student, also a senior lecturer at UNICEL, Madam Rosila Alias, who do research on probiotics and food safety. Okay, she tried to isolate probiotic and then try to apply and characterize the probiotics as the antibacterial, anti-cholesterol, attachment gene lactic acid, betrosine, and also peptide. So she's in the process to collect the data and produce the good results. So beside research, not only shared among the researcher or microbiologists, we also are keen to share with community. Okay, so at UNICEL, led by Dr. Haziq Azman, we have a group, we call us as STEM UNICEL, where we develop innovative learning module or kit or tool or materials that can aid people, especially the school children and also community, to polish their creative skills. Okay, and we focus to public. Normally we go to shopping mall, schools, teachers, and also policy makers to share, to create awareness about STEM, because currently, not only in Malaysia, I believe the whole world, interest toward science, technology, engineering, and mathematics decrease. So we try to help, and we're able to produce modules. And I tried to include microbiology in the module to give exposure to the community and also the school children. So this is one of our module, the STEM Curie module. Okay, this module has received a Gold Award in Malaysia Technology Expo 2020. So in the module, we have various content, and one of it is microworld. Okay, microworld is a basic activity about microbe. From the achievement of STEM Curie, we also develop many other specific kit, and I bring the microworld kit, okay, to Malaysia Technology Expo 2021, and Alhamdulillah, we achieve goal as well for specifically micro kit. So in the kit, we provide all the materials, and one of the activity is to grow yeast, and people can see the importance of microorganisms and related in bread making process, wine, and etc. So it is a low-cost kit, and we bring it to the school, to teachers, and community. During the pandemic, we also developed a STEM at home module. Okay, one of the module that I responsible is about STEM kitchen. So in this STEM kitchen also, I include the element of microbiology, because we try to include biology, chemistry, and physics. So for biology, I relate it with microorganisms. Okay, we try to give knowledge about why you wash your hands. So there is activity related to microorganisms. So, using STEM Unicell platform, me and my team try to inculcate knowledge, especially related to science, technology, engineering, and mathematics. And one of the component is about microbiology, to make sure community can know and gain their interest in microbiology world. So at the end of my talk, I would like to introduce some postgraduate opportunity in Unicell, because I already mentioned about all the projects that we do currently, okay, and we open for any candidate to do postgraduate program with us. Okay, inshallah, there is scholarship can be offered to the student. Okay, if related to microbiology, you can register student in Master of Life Science or Master of Science in Biotechnology or Doctor of Philosophy in Biotechnology. So if you are interested, you can ask me, or you can go to our website, Unicell in general, or CGS Unicell specifically, to get more information about all the programs. So with that, I end my presentations. So I hope we able to appreciate the microbiology world. We can see the applications of microbiology, because without microorganisms, we cannot continue our daily life or our daily activity. Okay, so that's all. Thank you. So I return back to the Master of Ceremony.
All right, thank you, Ibu Noraza Muhammad Nawawi. It was such an interesting presentation. Well, ladies and gentlemen, we come to question and answer sessions. I certainly believe that there will be many questions from participants, but this session will take only 30 minutes. And in these sessions, So for those who ask the question will be directly answered by Ibu Noraza, and then the rest also, will be directly answered by Ibu Noraza. And then for the participant who wants to ask the questions, you may raise your hand.
I think everything is clear.
Ya, Bapak Ibu, ladies and gentlemen, just raise your hand if you have any question to Ibu Noraza dealing with microbiology.
Rasanya semua sudah jelas. Sama ada jelas ataupun presentation saya tidak terlalu baik untuk menyampaikan maklumat.
Okay, Ms. Dian, maybe I will have a question for
All right.
Ibu Noraza.
Okay.
Bu Noraza, we know the COVID-19 that caused by coronavirus is more dangerous than AIDS syndrome and Ebola that spread from Africa, caused by Ebola virus, if I'm not make mistake. But we know the death rate of patient of Ebola more higher than the patient of COVID-19. My question is, what is happened? Maybe what have a relation about the character of virus, or about they spread in atmosphere or in human body, or what? Maybe coronavirus is very easy to get a mutation. Maybe I want you can explain about this. Thank you, Noraza.
All right, thank you, Ibu Sriwardani. Yeah, Ibu Noraza, you may directly answer the question.
Okay, Ibu, I think I'm not the best person to answer about this, but yes, there is differences. COVID-19, the death is higher compared to Ebola, though both are respiratory virus. Because about coronavirus, about maybe 3 million infected. But for Ebola, there is only, and it's only specific at more people, kill people in Africa. Because if I'm not mistaken, the Ebola virus is less contagious compared to COVID-19, and also the virus, the COVID-19 virus is easily mutate, and that's why we cannot guarantee we are protected from the virus.
Okay, Ibu Sriwardani, is it clear?
Yeah. Okay, thank you.
All right. Thank you. Maybe Pak Musin Khatib will ask question.
Thank you for the opportunity. I want to ask to Ibu Norazah Muhammad Nawawi, PhD, about the identification of bacteria. I have my research in identification of a bacteria in my bachelor study, maybe 10 years ago. I want to ask you, is the method of the identification of the bacteria that I have finished my research available to we use in the time? Okay, thank you.
All right. So the question is about the identification of bacteria. Okay, is it
Yes.
suitable to do to do the research of this bacteria in nowadays?
Yes, Pak Muhsin, may I know what kind of methods that you used before?
I use IMFIC method. IMFIC, indol, methyl red, and Voges prosceur.
IMPIC.
IMFIC, yes.
IMPIC method.
Okay, and at that time you were able to identify your microbes, yeah?
Yes.
Maybe I think that can be used as the what we can say preliminary before we can confirm using a recent technique, the molecular technique, sequencing to confirm the name of your microorganisms.
I use book what is yes, I use a book of the bacteriologist name.
Bergis material.
Yes, Bergis.
Bergis.
Yes.
Yeah, Bergis tu macam apa? The main reference. Dia macam kitab, memang kitab microbiology lah kan.
Microbiology.
Yes. So dia memang bagi based on the gram positive, gram negative, and also the cellulase fraction and everything. Yes, yes. It still can be used, but because we have current techniques which is more concrete or more specific, why not we better choose the current techniques, but yet we still can use that. Okay, my student currently also have still sometimes we kadang-kadang dapat, kadang tak dapat, so kita pergi juga tengok dia gram, tengok dia punya chemical reactions to confirm our identifications.
Ya, terima kasih Bu, sudah.
Thank you. And what's next? Any question?
Dicek.
There's one question from Muhammad Julian.
Bagaimana cara kami orang awam bisa mengambil hubungan dengan
berkaitan fungsi replikasi
DNA dengan PCR, Mikron atau Maksudnya mahu tahu sama ada dia Omicron atau bukan.
The relationship between DNA replication and PCR due to Omicron or COVID-19.
Fungsi replikasi DNA. Yes, memang dalam in PCR we use or we replicate the number of DNA. From the RNA we convert it into DNA and then we multiply it, and then from there they confirm that is it COVID-19 we infected or not. Certain lab it can detect up to whether it is Omicron or other variation. So it depends on the lab or request. Did I answer the questions?
All right, Pak Julian. I think it is clear. So next, there's one question from Pak Saleh Hidayat. The question is how to manage plastic wastewater as described earlier. Thank you.
Okay, basically, for batik wastewater is related to dye, because dye definitely will affect our environment. So in the research, we focus on the potential of microorganisms to degrade dye. So what we try is, we try to grow the bacteria in different kind of dye, and then after a certain period, we will see the dye, the color become faded, and then after when we know the bacteria, so after the application is either we use the bacteria itself or we use the enzymes. Okay, the dye degradation enzymes. Okay, we can choose. We try to try these two approach. So for bacteria, we try to grow in matrix. So this matrix, we hope we can throw the bacteria in the wastewater area, okay, and we hope the bacteria can degrade dye naturally. And at the same time, we also try to use enzymes, and then when we get the enzymes, we also will immobilize the enzymes. Okay, I mean we coat the enzymes, and we will try to use a similar process. But a lot of study related to enzyme, because enzymes quite sensitive to temperature, right? So that's how we plan and how to manage the batik wastewater.
So Pak
So
Saleh,
I answer my question.
what about the answer? All right. Another question from Pak Saleh. What kind of bacteria are used to reduce dyes, or what kind of enzymes?
Okay, currently our rhodococcus capable to degrade dye. Dr. Megela already tried to degrade safranin and few kind of a few kind of dye, okay, blue, red, green. Okay. Then currently we have rhodococcus, and in the process to isolate bacteria, other kind of bacteria, and related to enzymes, there are a lot of enzymes involved. For example, exoreductase, lactase, what else? Peroxidase. So they will try to try many kind or isolate those enzymes from the bacteria. So maybe the bacteria, one bacteria can only have one enzymes. So we can do consortium, means we combine the bacteria and the enzymes in order to try to solve the problem.
All right. Pak Saleh? Is it clear?
Ibu Sriwardani asked about
Yeah.
what rhodococcus.
Another question. How does bacteria or enzyme work for reduces the dyes?
Dye, yeah.
Yeah.
What kind of the bacteria? Rhodococcus.
Currently, rhodococcus. You asked me what kind of bacteria, right?
How does the bacteria?
How does it work?
How does the bacteria reduce the dyes?
How the bacteria
reduce the dyes.
We grow the bacteria with the dye, and after certain period of time, the dye become the growth become clear. And maybe day one it blue in color, and then after 24 hours or 48 hours it become clear.
Okay, thank you.
All right. Thank you, Pak Saleh. Okay, now next question from Ibu Sriwardani. What rhodococcus that you have isolated from indigenous or from where?
We have various species. We have ruber, we have different location. We isolate different kind of, not all different, but most. We have ruber. Tiba-tiba lupa nama.
Different species from different location.
Ya, ada juga yang sama, tapi
dari lokasi yang berbeda.
Lokasi yang berbeza. So, kalau dia we isolate from marine environment, so kita nampak dia potential more kepada hydrocarbon degradations yang contaminated dengan apa petroleum, for example.
Okay.
Because we tried palm oil, mill effluent, contaminated river.
River, marine, yeah. Okay.
We have few.
Yeah, okay. Thank you.
All right. Thank you, Bu Sri. Another question? The participant can raise their hand or write down the question on chat room. All right, Ibu Marlina or Mas Denisa will ask the question. Come on, Ibu.
Okay, thank you for opportunity. I would like to ask about how to rely about high technology. We know as a research, sometimes we confuse if we have children or we have as a teacher, not as a research, how to relate in children to know a bacteria. I have a experience use hands wash hands like you research, but you have a good idea. Make a micro word, yeah, micro word, and please share about a micro word because maybe in the other time I will use this method to make tools in my class, because I like microbiology, but sometimes I confuse to relate it about my research in my children or other as a and now about the microbia. Thank you. Maybe just a share about the micro word. Thank you.
Okay. Okay, basically in microworld kit, one of the just now I mentioned that we ask them to grow the yeast, and the kids can see how it's grow. At the same time, we also have activity where you can wash your hands. Either you wash or you do not wash your hands and put it on bread, for example, and then you keep the bread for 24 to 72 hours and then observe what is growing on the bread. Or you can use a simple agar, kita buat jelly biasa saja kan. Okay, that's also you can use it. You put it under your sink, for example, your cupboard, table, and then you can see the bacteria is growing. That's a simple activity related to microorganisms that you can do. Other than that, you can maybe, if more interested, you can buy some simple microscope. You can buy from the online platform. So you can make your children to observe the water from tap water, from the drain, or from river. So inshallah, they will observe different kind of microorganisms from the environmental sample.
Thank you. How long we use your micro word? Or just 1 hour or one day?
If you want to grow? You mean if you want to grow?
I want to know. You make a micro word.
We have the kit actually. We have the kit. We sell the kit. Inside the kit, we have all the materials. So we sell the kit. So people can buy the kit. It's a complete kit where they can just take out the materials and do the activity.
This is special kit. You make a kit. Oh.
Yes, we develop the kit.
Okay, thank you for information.
All right. Thank you, Ibu Marlina. So I think we still have 10 minutes more. Any questions? Kita punya 10 menit lagi, Bapak Ibu. Jadi kalau ada pertanyaan, silakan. Right? Any question? Mhm. Yeah, Bapak Ibu, you may raise your hand or write down on the chat room if you have any questions. Still have 10 minutes more. Yeah. All right, Pak Eri maybe. You
I will
raise
try.
hand.
Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam.
I have a question in my head. Why the AIDS virus vaccine hasn't been found yet? Coronavirus, COVID-19 have a vaccine. The vaccine is we know three of kind vaccine in the world, but why the AIDS virus vaccine has been found yet until now? Maybe you can tell for our about this question.
All right. About the vaccine. Okay.
Yeah, they tried to, okay, but because HIV is affect the immune response, right? So there is difficulty in the development of the vaccine, because normally a vaccine will mimic the natural immunity. Okay, but for AIDS, it cannot do that. And then because I think it's related to the immune system. And then the structure of the virus itself, okay, is quite complicated. So that's why they still have the clinical trials to develop the HIV vaccines.
Okay, Pak Eri, is it clear?
Okay, thank you, Miss Dian. Thank you, Bu Nuraza.
All right. Any questions? We still have 5 minutes. Ya, Bapak Ibu, ada pertanyaan lagi? Silakan. Kita masih punya waktu 5 menit. All right. So, one question from Pak Saleh Hidayat again. Is Rhodococcus easy to breed? Is it highly pathogenic?
Rhodococcus not as easy as E. coli. Okay, if E. coli between 24 hours you can get high OD readings, but for Rhodococcus, certain species after 24 hours also we cannot see the orange color, and Rhodococcus is non-pathogenic except one species which cause disease to animals, Rhodococcus. But other than that, it's not pathogenic. So that's why we use it. But the best about Rhodococcus is it is robust, okay, because it has a strong structure. So that's why it can resistant toward many kind of pollutant. So it's not pathogenic except one Rhodococcus. Rhodococcus is a zoonotic. Rhodococcus equi. It's an animal pathogenic Rhodococcus. But other than equi, it's non-pathogenic.
Alright. Ada lagi Bapak Ibu pertanyaan di questions? Still have 2 minutes. Yeah. Or is it clear all?
Is it clear? I think.
All right. Yeah, I can conclude that, yeah, if it is clear, I can conclude that the microbiological research is currently widely applied in the industrial world. And it becomes an interesting topic to discuss, yeah, both for general public, yeah, or the industrial community. And it also an interesting subject, yeah, for lecturers and teachers, yeah, and particularly for graduate students of biology education study. All right. So, well, ladies and gentlemen, finally we come to the last agenda. I would like to say thank you very much for keynote speaker, Ibu Noraza Muhammad Nawawi, that has presented an interesting presentation. And all of the audiences, ladies and gentlemen, for actively participating in this webinar. All right. Hopefully, this webinar will give many benefits for us. All right. So, thank you for your attention. Before we leave, let's the committee or the head of biology education study program will give a certificate to keynote speaker, and also the committee will take the picture together, yeah, for the participant on this Zoom webinar. All right. 1, 2, 3. Again. Once more. 1, 2, 3. All right. Thank you for your coming on this webinar Zoom. I want to say I'm so sorry if I have so many mistakes in guiding this webinar. All right, thank you for your attention. Thank you Ibu Noraza Muhammad Nawawi. Thank you Ibu Sri Wardani. Thank you Pak Eko. Thank you Pak Saleh. And then all the participants, ladies and gentlemen, I close this webinar Zoom by Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Alhamdulillah, terima kasih banyak Ibu Noraza.
Sama. Harap memenuhi ya.
Sudah sangat membantu ini untuk pemahaman mahasiswa graduate kita. Ya, mudah-mudahan di lain waktu bisa terus bekerja sama untuk saling memberikan ilmunya, Bu Noraza, untuk kita semua.
Mungkin ada idea untuk kolaborasi research ke pun boleh diutarakan.
Iya, oke, alhamdulillah. Ya, insya Allah nanti Ibu dalam waktu dekat bisa menghubungi Bu Noraza.
Insya Allah, boleh.
Terima kasih Bu Noraza.
Ibu tertarik dengan STEM.
STEM itu memang sangat menarik.
STEM Unisel ya.
Kita banyak aktiviti.
Iya. Apalagi outdoor lab-nya tadi. Keren. Ya, oke. Terima kasih semuanya. Assalamualaikum.
Waalaikumsalam.
Izin break semuanya ya. Assalamualaikum.
Ya, terima kasih Pak Saleh.
Terima kasih Pak Saleh actively participating.
Terima kasih Bu Noraza.
Saya keluar.